Good morning to all the participants. I hope they're all teachers here in the 2 weeks refresher program. As the title suggests, I will talk about a class of proteins named hemoglobins. To me, nothing signifies the rich cultural heritage of our monuments in our motherland more than the imposing red fort. In the world of proteins, nothing is more imposing as the red colored protein we all know as hemoglobin. It's a fascinating protein. We all know that it's very important to life itself because it transports oxygen from the lungs to the tissues and brings back carbon dioxide to the lungs to be expelled. The red color of the protein comes from a heme group, which is a cofactor, a prosthetic group present inside the protein. What I'm going to do today is tell you new developments in the field of hemoglobin. This is a textbook molecule, right? It's very well studied at the undergraduate as well as postgraduate level. So one would wonder what is new for me to talk to you about. But I thought this is a refresher program and where you update your knowledge in a specific field. So this is an area where you can know about the new developments, which are constantly pushing the boundary, busting a lot of myths, changing concepts about the protein itself. And I wish to impress upon you at the end of the talk that these are very important molecules which needs to be studied more and more in more details. Okay, and you all know Linus Pauling, one of the finest biochemists, who not one, but 2 Nobel Prizes. One for his work with peptides, talking about secondary structures of peptides, and the second was a Peace Prize. Pauling, at one point of time, had said that hemoglobin is in some ways the most interesting of all substances, and I do believe in the same. One major area of research in my lab is working with hemoglobins, and I'll tell you why. And it has been part of folklore and part of designs, installations. If you go to the US, you will see such structures on the streets, even where this brown colored molecule represents the heme and the oxygen that binds to the protein. This is another reason why I wanted to talk to you about this. I keep telling our teachers that hemoglobin is a good protein molecule to do a lot of practical work in life sciences laboratories. And with the new education policy, where research is an integral part in the 4th year, these molecules are very amenable to work in the laboratory, especially because of their color. And you can easily demonstrate to students, how proteins work. When you express them in E. coli, for example, bacteria, you'll see a very red pellet because of overexpression of the protein. So you know that expression has worked well. You don't need to do anything. Visibly, you can see. And you can grow them in kilograms. I have used very large fermenters to make kilograms of red proteins, which can be stored in the freezer for years and can be worked on at any point of time that you need. Once you lyse the cells, you'll see a very red protein, so you know where your protein is. If you do column chromatography, you see your protein running over the column, so it's very easy to demonstrate the principles of column chromatography to students as well. If you crystallize, you'll see a red colored crystal. So that's the reason I call this class of proteins the Red Goldmine. I don't need to talk about this much because you all know what their function is: oxygen transport and storage, okay, where they carry oxygen from the lungs, release them to the tissues where myoglobin takes them up, and then hemoglobin carries oxygen, carbon dioxide back to the lungs where it is expelled. I just want to draw your attention to the simple chemical reaction that hemoglobin does. It binds to oxygen in a reversible reaction, forming oxyhemoglobin, which can also dissociate back into hemoglobin and oxygen. This is a constant, dynamic equilibrium. Okay? And the rate of reaction between hemoglobin and oxygen is signified by the on rate, or sometimes called the association rate. And the dissociation of oxyhemoglobin back to hemoglobin and oxygen is often called the off rate or dissociation rate. These terms will be used in my talk, so I thought I'll just tell you about them. This is also textbook knowledge. This is taken from this textbook by Dickerson and Guys. Hemoglobin consists of two parts, as you know, a polypeptide, which is called the apoglobin, and the heme prosthetic group. The structure of heme is shown in details here with iron at the center. And you have the chiral rings, 4 nitrogen, satisfy the coordination state of iron, okay, which can be both in the ferric state and the ferrous state. And as you know, oxygen binds in the ferrous state. Okay? And this combines with the apoglobin to form the hologlobin or hemoglobin, which is red in color because of the interaction of the heme with amino acid side chains in the protein, right? This is a classical picture, a three-dimensional structure for hemoglobin. Okay, it's an all alpha helical protein, so there are no beta sheets as secondary structures. The only kind of alpha helices that you see in hemoglobins are alpha helices. Usually, there are 8 alpha helices, and they are numbered A to H, starting from the N terminus. So this is A helix, B helix, right? C helix, it comes back. See here, C helix, folds here, D helix, E helix, folds back, F helix, G, and H helix. And H helix is the red one. Okay, so A to H, usually there are 8 helices. And they have pockets here on the top of the heme and the bottom of the heme, which are called active site pockets. Heme is more or less a planar molecule, and gaseous molecules like oxygen, carbon monoxide, nitric oxide can bind heme, and hemoglobin has learned to discriminate oxygen, so it binds oxygen better than carbon monoxide and nitric oxide under physiological conditions, though normally hemoglobins has a very high affinity for carbon monoxide as well, which results in carbon monoxide poisoning and stuff. But this is what is a classical picture. This is macroscopic view of the active site. As I said, this is the iron in the heme. Okay, you have one histidine, okay, which covalently binds to the iron. This is the only covalent linkage between heme and the peptide. Okay, the rest is peptide. This histidine is called F8 because usually this is the 8th residue on the F helix. And then on the top of it, you have, this is called the proximal side of the pocket. This is called the distal side of the pocket. Classically, you have another histidine, which is the 7th residue on the E helix. Okay, and it forms, there's a water molecule here in the pocket. And when there is no oxygen bound, this histidine hydrogen bonds with this water molecule. Okay, this is classically known. This is all established textbook knowledge. Okay? And the molecules that we know in the hemoglobin superfamily, one is the hemoglobin, the iron-containing oxygen transport protein in the red blood cells of vertebrates, and another is myoglobin, which is a monomeric cousin of hemoglobin. Hemoglobin is tetramer. It's important for storage, found in muscle tissues. Muscle tissues is all red in color because there are millimolar concentrations of myoglobin that is expressed here. And this kind of proteins are called classical hemoglobins or traditional hemoglobins. They are textbook knowledge. Okay? Hemoglobin and myoglobin have also been important historically okay, because of because of various landmarks, okay. They were the first protein. Myoglobin was the first protein for which three-dimensional X-ray structure was determined. So its three-dimensional structure was determined for myoglobin. That's the first protein. The first protein to have its mass accurately measured, first protein to be studied by ultracentrifugation, first protein to be associated with a physiological condition, especially a pathogenic condition, sickle cell, first protein to show that a point mutation can cause problem, that is anemic cell, sickle cell hemoglobin. And also, sorry, cooperativity, the theories of cooperativity, how ligand binding, first, the phenomenon of cooperativity actually was studied with hemoglobins. There are classical hemoglobins elsewhere other than red blood cells, okay, and the tissues that we talked about. Plants at one point, in the early 1900s and 1930 or so, plant hemoglobins were discovered, especially in the soybean root system, for example, where root nodules are formed. Okay, and when hemoglobin is expressed, when they are infected by rhizobia, you see that the protein is expressed in very high quantities, millimolar quantities. This is a cross section of a root nodule showing that hemoglobin is expressed in very high concentration. Their function is also transport and storage in plants. So the hemoglobins were at one point thought to be found in limited organisms, some plants like this, and maybe vertebrates. However, what is important, what made hemoglobin so popular, where over the years it was found that they are ubiquitously present across all life forms, archaea, okay, bacteria, eukaryotes, all life forms. And these discoveries were specially made due to extensive genome mining, okay, where putative hemoglobins were identified. This is a little bit of an old data, which shows that 2,200 bacterial, 140 archaeal, and 67 eukaryotic genomes were identified. Okay, and there were different various kinds of hemoglobin that were identified as well. As you can see, they could be only hemoglobin domains, as the classical ones that we talked about, or they could be associated, hybrid domains associated with other kinds of domains. I'll talk about in a minute. So this is what made hemoglobins so interesting. At one point of time, it was thought that we know everything that we ought to know. We know their function, we know their physiological importance, and it's textbook knowledge. But with such discovery, it was realized that hemoglobins must have been more important in evolution, that they are present in all kinds of organisms. You can name any organism today, and hemoglobins are present there. Okay, that made things very important for us. Just to name a few, okay, and also ones that we have worked upon. For example, an algae, Chlamydomonas hemoglobin, it was found in rice. It was found in the cyanobacterium named Synethocystis, in Drosophila, in Leishmania, which causes leishmaniasis, Kala Azar, in Arabidopsis, and in humans also. In addition to hemoglobin and myoglobin, one new hemoglobin was discovered, which are localized in the brain and in the retina, associated with neurons. It was called neuroglobin. And when neuroglobin was discovered, this was a Nature paper, people got more interested. That all this while we knew only about RBC hemoglobins and myoglobin, and yet there is another hemoglobin in humans, which is called neuroglobin. So all this came into light, and we worked on all of them. Okay, but we made specific discoveries for these two in the lab, Clemmy hemoglobin and Leishmania hemoglobin, when it was not known to anyone. Okay, and these hemoglobins differed from the classical. We call them new hemoglobins or sometimes novel hemoglobins, which are very different from the classical hemoglobin in their ubiquity. As I told you, they are ubiquitous. Multiplicity, they are found in multiple numbers. I'll just show you examples. They varied in their sequence quite a bit. Sometimes you will end up finding only 10 to 15% sequence similarity with the classical hemoglobins. The amino acid length also varied. Usually, classically, hemoglobins contain approximately 150 amino acids, but shorter and longer hemoglobins were discovered. They had lot of conformational plasticity in their structure, which I'll show as an example. Active site diversity, their kinetics, that is ligand binding, also were different. They differed in their stability or cellular localization and function. There are a lot of functions that are talked about. Okay, in some cases, they have shown to not to be transport or oxygen proteins, but have various other functions, which I'll also talk to about. The questions were, do they have common functions? Do they have multiple diverse functions? That's what makes them so interesting. Okay, so if I compare to cricket, which is a religion in the country, the classical hemoglobins would be like test cricket, right? And when these new hemoglobins or novel hemoglobins were discovered, they were like one-day cricket, a new form, which is much more exciting when it was first innovated. One example, as I said, that we had discovered in our lab sometimes back is in Leishmania, and from genome mining, and we see that when we did a modeling for the protein, it folded into alpha helices, typical of a hemoglobin. It had heme, but very interestingly, immediate difference what we observed, as I told you, that classical hemoglobins have a histidine at the bottom and a histidine at the top, but this protein had a leucine at the top, okay? Which was, we don't know why. Usually you have a histidine, but this one had a leucine in the pocket at this position, okay, which is very interesting. But before we could do more work on it, as it always happens, science is very competitive. This protein was reported by someone else, but I'm happy that it was reported from India, in Kolkata. They published this paper before we could do, and they showed that it is an oxygen sensor, not really an oxygen transport or storage protein, but as an oxygen sensor, which triggers adenylate cyclase and helps in a lot of, has regulatory role and prevents cell death during hypoxia. So we did not do much with it. Later on, we discovered hemoglobins in a green algae called Chlamydomonas. And as I told you, what we immediately noticed by mining the genome was that they were present in multiples. These Chlamydomonas, we found that there are 12 globins, in this protein. There are 12 globins, and they are different kinds. Some consists of the globin-like domains. Other will consist of one globin domain, and there are other parts of the molecule. Some of them did not match to any known protein. Okay, so these are kind of hybrid molecules, okay, with other domains as well. And this continued. We did not discover this, but other scientists discovered that C. elegans, which is model nematode, as you might all know, it's a model organism, which is studied because many of its genes are similar to humans, has as many as 33 globins. This was mind-boggling to us, okay, that they have 33 globins. Okay? And it's a microscopic organism. Drosophila, which is another model, okay, for genetics and all kinds of studies, also had 5 globins. And later on, I managed to form a consortium in South Campus, and one of our colleagues from genetics department, Dr. Sujit Sarkar, worked on many of these globins and published wonderful papers on their various functions. Now, in terms of location, classical hemoglobins, as I told you, are mostly in RBCs, in muscles, and in plants, sometimes in roots. But these new hemoglobins were found to be cytoplasmic and there are a few reports of them being present in the nucleus as well. Okay, so when they're present in the cytoplasm, the nucleus, they are obviously going to have a lot of diverse functions in addition to oxygen transport, storage, if any. And even in chlamydomonas, we saw membrane association of some of these globins. Out of these 12, some globins showed membrane association as well, which was completely new knowledge, not known in the textbook. Okay. In terms of their oligomeric state, classical hemoglobins are either monomer like myoglobin, or they are tetramer, right? 4 polypeptide subunits like in the hemoglobin. And in some cases, in Earth-1 and others, they were found to have as many as 22 subunits, very complex molecules. Okay. But most of these new globins or novel globins were either monomer or dimer. Okay. We did not see any higher order oligomer. Okay, mostly monomer, and some of them are dimer. Protein folds, okay, and protein folding is very important for its function. Classically, if you open up any textbook, you will see that hemoglobins have a very typical fold, which is called 3-on-3 alpha helical globin fold, which means that out of the 8 helices that I talked to you about, 3 helices are in one plane. Okay, as shown here, this A helix, E helix, and F helix. Okay, they are in one plane, while B, G, and H are in another plane. Okay, so they form these sandwich shown here. That's why sometimes they are also sandwich globin fold. Now, in this, very interestingly, okay, it was found that they are 2-on-2 helical globin fold. So instead of 3, now you have 2 helices, the B helix and the E helix, and G helix and the H helix, which formed 2 planes, and the heme was in between those 2 planes. So this was called a 2-on-2 alpha helical globin fold, very different from the 3-on-3 alpha helical globin fold. And in some cases, in the new hemoglobins, some helices were often not there; they were also deleted. Okay, so this is how they differed. Okay. As I talked to you about the active site, I showed in Leishmania there is a leucine which is already different. A very interesting observation was also found, in some of these new hemoglobins. If you look at the active site, okay, in and around the heme pocket, okay, you see for the classical hemoglobins, okay, as I told you, you have one histidine which is covalently linked to iron, and you have another histidine which is not linked to iron, but it is there, and when oxygen binds, it forms a hydrogen bond to oxygen and stabilizes the oxygen here. That's the role of this histidine, okay? And that's how oxygen binds here. So the histidine is very important for that purpose, okay? And as I told you that we have an iron at the center of the heme molecule, and as you know, iron is 6 coordinated, and I showed you the structure of heme. 4 nitrogen satisfy its coordination, and one of them, the 5th one, is covalently bonded to a histidine through nitrogen. So the 6th one is open for ligands to bind, which satisfied the 6 coordination chemistry of iron. In these new classes, okay, the novel hemoglobins, the new hemoglobins that we found, we were surprised to see that this is hexa-coordinated, okay, just like cytochromes or P450s. Histidine from the bottom, histidine from the top, both are covalently linked to iron. Okay, these classes of hemoglobins were called as hexa-coordinated hemoglobin. Okay, so this came as much of a surprise, very interesting, and intriguing. And just as I talked about cricket, so we ended up discovering 2020, from going from classical test cricket to 50-50 now, 2020. So new information. Okay, this type of hemoglobins were called hexa-coordinated. You can always ask me that how do we distinguish? Very easily. If I give you a hemoglobin, whether it's penta-coordinated and hexa-coordinated, it's very easy to do using spectroscopy. For example, simple absorbance spectroscopy. A pentacoordinate hemoglobin will give, this is the typical absorbance spectroscopy. If you measure absorbance in the visible region, you will see a peak here called the Sore peak, and a peak in the visible region here around 450, and there's a single peak here. If you do another form of spectroscopy, which is called EPR, electron paramagnetic resonance, where you study the properties of electrons in a very high magnetic field, it will give this sort of spectrum. When you have a hexacoordinated protein, we immediately saw that there is a difference here. Instead of a single peak, you get two peaks here because of the strong covalent binding. And the EPR spectrum is very, very different from a pentacoordinated protein. Now, whether it is because of this coordination, how do you verify? In life sciences, there's one technique called mutation analysis. You do a site-directed mutagenesis. So we mutated histidine to something else, to a side chain which is not histidine, so it cannot bind to iron, for example, leucine. And we saw that this spectrum and this spectrum goes back to this spectrum. Okay, for example, this is E7L rice hemoglobin, where histidine is mutated to leucine, and now you lose these two peaks, you get back one peak, and the EPR spectrum also becomes like pentacoordinated protein, okay, which clearly proves this. Now, when we studied cyanobacterium protein, I crystallized this protein and solved the crystal structure as well. We saw that it is indeed a hexacoordinated protein. You have one histidine at the bottom, one histidine at the top. In addition, what came as an absolute surprise for us, we saw a third histidine, okay, which binds to propionate group here, a vinyl group here, in the heme. Forms a covalent linkages. So there were three histidine linkages in this hemoglobin from cyanobacteria called the Synechocystis species. So this hemoglobin is called Synechocystis hemoglobin. And this was an absolute surprise. Okay, so you end up, watching 10 overs cricket now rather than 20 overs cricket. It is so interesting and so intelligent. Okay, there are three histidine. And naturally, this protein was found to be very, very stable. I have studied stability of these proteins, published papers. It's very interesting. You go to pH 2, you use guanine hydrochloride, urea, high temperature. You cannot get the heme out of this protein. For classical hemoglobins, heme can easily be taken out from the protein. But for this protein you cannot, because of three histidine link. This is an absolutely new knowledge, not there in textbook chemistry. What about various habitats? Okay, for example, extremophiles. So we looked for the same, okay, and we found one hemoglobin. We have not published this yet, from thermoacidophilic, red algae, okay, which grows in thermophilic and kind of an acidic environment. It's called, this algae is called Galdiraria sulfuraria, and we found this hemoglobin in this organism with typical fold and heme also. And when we did, we studied its stability. Okay, we cloned the protein, expressed, purified, and studied the stability. Very interestingly, we found that stability, for example, 51, is much less than myoglobin, which was stable up to 77, or the green algae. So this protein, this hemoglobin from a thermophilic organism, was less stable than mesophilic hemoglobins. We still do not know why. We have done a lot of studies and a lot of analysis, but still we don't know. There are certain structural features that we are close to being identifying, for example, some GG motifs and some loops which might cause this, some hydrophilic specific electrostatic interactions, but this came as much of a surprise. Okay, and as I told you, that oxygen goes into the molecule. Okay, and usually this mechanism is very well known and sometimes is available in classical textbooks as well. That histidine here in the distal pocket acts as a gate, and this moves away and let oxygen come in and bind, and then histidine will hydrogen bond to that oxygen. Okay, and that is shown here in a better way. This is a classical textbook knowledge. But in case of these new hemoglobins, novel hemoglobins, okay, which were sometimes shorter in length also, we call them truncated hemoglobins, they showed a very interesting characteristic feature, which we call tunnels, as shown in gray here. Come in and bind. This is hemoglobin from Mycobacterium. Okay, so this is shown to have tunnels in their protein, as shown here in this magenta color. They have long tunnel. Okay, they have hydrophobic side chains in the tunnel, and maybe they help in migration of oxygen or storage of oxygen inside the protein molecule. These tunnels were discovered. Okay, which is another unique property. You have heard about conformational changes. There's a lot of dynamics in hemoglobin molecule. For example, this is a deoxyhemoglobin found in red blood cells, which transports oxygen. You see there are 4 molecules in different color because this is tetrameric. Heme is shown in red. When it binds oxygen, this red ones here are the oxygen. If you toggle between this, hopefully you can see there is conformational change between oxy and deoxyhemoglobin. You see here, this is a little puckered. In oxyhemoglobin, they straighten up to bind oxygen, and there are small changes in the molecule. These conformational changes are known. But when we saw this new hemoglobin, for example, syncytial hemoglobin, okay, where I told you there were 3 histidines, we saw remarkable conformational change, which was not seen in hemoglobin or myoglobin, especially when it binds oxygen or any other ligand. For example, this histidine here, which is in the pocket, when ligand binds, this moves away totally. It goes here. This is a huge movement for protein. Okay? And this helix here, it totally moves away. Okay? You see, this is total displacement of this helix, and this histidine also goes away. So such conformational change, which is maximum in these new hemoglobins, is not required for oxygen transport storage. So what kind of function can they do? For example, can they do ligand sensing? When they sense the amount of oxygen, levels of oxygen in the body, because later on they were found to be overexpressed during hypoxia. Okay. So as you can see, I showed here the conformational changes in red and gray. All this E10 moves here, all these residues, E11 moves here. Lot of conformational change, which is unique for these proteins. Maybe it was thought that they have a role in signaling with such kind of conformational change. Can do signaling. Lot of plants were found to hemoglobin. For us, we were the first, from India to solve the 3-dimensional structure of a hemoglobin from Arabidopsis thaliana. Arabidopsis thaliana have 3 classes of hemoglobins, hemoglobins 1, 2, and 3, and we solved this structure, and we found that it is a dimer, it is hexa-coordinated, okay, and it has tunnel as well. Okay, so this is a plant hemoglobin, so the obvious question was, what roles do such tunnels play? When we compared this with literature, we found that there is one class of hemoglobin known, which are flavohemoglobins, which have a globin domain, and they have, in addition to globin domain, they have another domain, which is an FAD binding domain, and they act as nitric oxide dioxygenase. That is, they destroy nitric oxide, because nitric oxide can be very toxic if produced in high amounts. Okay? So they reduce nitric oxide to nitrates and protect the protein. And these flavohemoglobins also have some kind of a tunnel. So our hypothesis was that these new hemoglobins with tunnels could be functioning like flavohemoglobins and may have nitric oxide dioxygenase activity, so it will be protective in their function. Okay? We also crystallized a truncated plant hemoglobin. Truncated means they're shorter in length. Okay, and we found that this hemoglobin have N and C terminus, which are totally disordered. They keep moving, okay, constantly, unlike any other hemoglobins. So, the question is, what is their function? Maybe they use this very flexible, mobile N and C terminus to look for changes, some kind of signaling, bind to other molecules. So they have other partners that they bind to and do their function, which is a possibility. Okay. Again, when we looked at, I told you about K on rates and off rates. When we looked at those numbers, which is called a kinetic analysis, that is the rate of binding of ligand between the classical pentacoordinate and hexacoordinate molecule, you see the of affinity for oxygen are this kind of numbers for myoglobin, for human hemoglobin, okay, for the plant hemoglobin. But when we observe this, hexacoordinate hemoglobins, you see the affinity is much, much higher. Okay, so they bind oxygen with much, much higher affinity. Though they have a histidine sitting in the pocket which binds to iron, yet they bind oxygen with higher affinity, which means that histidine in the distal pocket moves away, okay, allows oxygen to bind. And once oxygen is bound, they bind with a very high affinity. Okay, with such high affinity, they cannot function as oxygen transport or storage protein, because they need to release the oxygen also, okay, as per the functions, classical functions of oxygen transport protein. If they bind it too tightly, they cannot do that. That's also a new finding for all these kinds of hemoglobins. If you see, they're very different. They're even for carbon monoxide, their bindings are very, very different than the classical hemoglobin. So they may not function as oxygen transport or storage protein. They may function as sensors, as scavenging molecules, because they have nitric oxide dioxygenase function or some kind of a signal transduction. Okay. In terms of cooperativity, that's kind of a movement that you observed in hemoglobin. When ligand binds to one subunit, the second subunit binds oxygen with higher affinity, and subsequently third and fourth subunit keeps on binding with higher affinity, which is a function called cooperativity. And this is the conformational change that they show, as you can see here, between the oxy and the deoxy. Okay? But as I told you, synechosis shows much more, much higher conformational change, much more movement in the molecule. And when their binding affinity curves were observed, this is again a textbook knowledge. You see a sigmoidal oxygen binding curve for myoglobin, a rectangular hyperbolic curve for myoglobin, and a sigmoidal binding curve for human hemoglobin, right? So you have rectangular hyperbola versus a human hemoglobin, which shows a sigmoidal binding curve. Okay, but when we saw, and their oxygen affinity usually in the range of 1 to 10 micromolar, and their dissociation rates is 1 second inverse, and hemoglobin is present in 1 millimolar concentration. So this is a requirement for hemoglobins to act as oxygen transport proteins. Okay, but when you see these new proteins, okay, they showed very different kind of binding curves. Okay, they are not hyperbola or sigmoidal in nature, and sometimes they have all these different kind of phases. And as you can see here, 3 phases instead of 2 phases that you see. They have very high oxygen affinity, and their kinetics is very different. And the amount of these proteins, found in organisms, where in sometimes nanomolar or micromolar range, unlike the hemoglobins which are found in molar range. Now, to act as oxygen transport protein, the concentration of hemoglobin must be higher than dissolved oxygen. Dissolved oxygen concentration goes as much as 262 micromolar. Hemoglobins are present in millimolar quantities, so they can bind many more oxygen molecules and help with diffusion. But the new hemoglobins were in the range of nanomolar or micromolar. So obviously, and their kinetics are all different. So obviously, they cannot act as oxygen transport proteins, or maybe they add. In addition to that, they may have other functions like heme binding, storage, and transport, as nitric oxide dioxygenase, nitric oxide sensors, and tumor suppressor protein as function was associated with them, stress response, electron transport, etc., etc. As you can see, some of the publications here. I'm not going into detail, but cytoglobin, tumor hypoxia, again from South Campus, because 7 groups formed a consortium to work on hemoglobins. Rise phytoglobins regulate response under low mineral nutrients and abiotic stresses, in Arabidopsis. Or they can have some defense, against, a pathogen attack, which were all done in, Drosophila is required for development and oxidative stress response. Okay, so they have all these properties which we published. So these are totally new knowledge about hemoglobins. I'm sure you'll find them very interesting and intriguing. And even a lot of bioinformatics study in computer studies can be done with these proteins, looking at their genomes and mining the genomes and looking and modeling the proteins, etc. To end this, we talked about new aspects of hemoglobins. To end the talk today is what else can you do? Why to study hemoglobins other than those interesting properties that we talked about? Is they can have various other applications. A lot of translational research can be done. When I was in US, a cosmetic company contacted us, and we made hemoglobins in liters, several hundred liters, because they wanted to try them in cosmetics, especially the plant hemoglobin from legumes called leg hemoglobin, because they have a large pocket and they can scavenge a lot of free radicals in the skin. So the skin can get rejuvenated. That was their idea, their concept, if they use hemoglobin in a personal care product, like moisturizers and creams. And I have not followed up that now, but they have been purchasing and doing a lot of studies on skin models, etc. Okay, they can be useful as carbon monoxide and nitric oxide sensors, and also as blood substitutes. Okay, as I said, we made so much hemoglobin for this company, and they have importance for sickle cell anemia, which I suppose that's a totally different project I'm working on. I'll not talk about today. As you know, with this mutation present at this 6th position in beta hemoglobins, beta chain, you end up getting sickle cell hemoglobins, which do not transport oxygen well, and they cause a lot of pain and a lot of suffering for patients, sickle cell anemia. We also discovered, as I said, that the hemoglobin in the brain, which is called neuroglobin, that they form amyloids. I am sure you've heard this term, and that was a new discovery for us. Amyloids are a kind of fibrils or aggregates that they form within the cell. These are some TEM images that we showed, okay, and which can have implications in neurodegenerative diseases. Okay, amyloids, as you know, form these beta structures, fibrillar, without much branching. They are not soluble, they're insoluble, they deposit, and they cause a lot of neurodegenerative diseases. You might have heard about Parkinson's, Alzheimer's, which are caused by proteins that aggregate. And neuroglobin was found to have this property. We discovered they are found to have cytotoxicity. We used human neuroblastoma cells, did an MTT assay, and found that these proteins could be cytotoxic. Okay, they can destroy this. These amyloids can destroy the cells. Very interesting aspect that we observed, and they also cause problems in channels, in electric conductance. We did this work with IIT Madras. Okay, So they may have neurodegenerative implications, is what we are saying. Okay, we published this paper as well. And I'm not going to talk into details. You can use hemoglobins as blood substitutes because blood is in short supply, and blood is supplied by transfusion, which is obtained by donation. Especially for surgery, battle wound, etc. We have shortage of blood all the time. As you know, so many blood camps are held all around, but still we have shortage. Okay, shortage of 40 million units every year. Okay, and there are various reasons for shortage, disadvantages of donated blood, which do not work well because of shortage, poor shelf life, storage problem, transmission of disease when you take blood from one person to another person, blood type mismatch, etc. So one way that people think this problem we can get rid of, using substitutes, blood substitutes, which mimic the functions of the blood only on short term, 4 hours, 6 hours, not long term. For long term, you may need stem cell based options, but short terms. And there are certain blood substitutes that have been tried. For example, fluorinated hydrocarbons, PFCs, have been tried. But what is more important for us is hemoglobin-based oxygen carrier. Can we use hemoglobins to carry oxygen? Just the protein, make lots of the proteins in the laboratory and use it as a transport protein, which is called an hemoglobin-based oxygen carrier. We have been working with this. It will have a lot of advantage. No need to check blood types, long shelf life, free of side effects or disease transmission, cheap to manufacture. Okay, and they can be used as an alternate. We are trying this to solve this problem, and we are not the first one in this. I must tell you that there have been several labs, especially in the US and elsewhere in the Europe. People have tried to use hemoglobin as an oxygen carrier, and they have been largely successful, especially when using them in cattle and animals. But one problem was that whenever you use hemoglobins outside their RBCs, they will release heme, okay, when you inject them. And this free heme is very toxic, okay, and it will cause a lot of other problems. Okay, so one problem that is not solved is, can we prevent heme coming out of hemoglobin? So that means to improve the heme stability. And when I was doing this, that is new, which we did for the first time. We said, let's learn from nature. And I already told you about this protein from cyanobacteria called Synthesis hemoglobin, which had 3 histidines. And from this protein does not release heme. Heme doesn't go out of this protein. So if you can engineer this stability in hemoglobin, okay, that is, introduce a histidine in hemoglobin so that it binds heme and heme is not released, right? So when you use that kind of hemoglobin as blood substitute, then there will be no toxicity. Okay, that is what we did. And I'll stop here. I'll not explain, but we did this. We used myoglobin first, which is a single chain, single subunit, small protein. We tried to do this, chain this heme to the histidine. Okay, and you all know this. I don't have to explain this statement thundered by a legend of Indian history: "Give me blood, I will give you freedom." For us, we are taking it in a different way. Okay, we reduce or curtail the freedom a little bit to get blood substitutes, and we tried this. I'm not going to explain the details, which is not required. In myoglobin, and we found that in myoglobin that this heme is released, and it goes into the if you take this at low pH, okay, the heme will be released. And if you add an organic solvent, heme will go into the organic solvent, protein will stay in the aqueous solvent. Okay, but when we made this new mutant, okay, which is I107H, we found that heme stays in the water, which means that it is inside the protein, it is not released. So we successfully engineered this, did a lot of other studies to prove that yes, this protein is more stable. You see temperature also. This protein is more stable. And we did a technique called differential scanning calorimetry, which shows that that mutant has higher stability. Okay, as shown here, up to 85 degree, it can be stable compared to the wild type. Okay, and it binds oxygen also well. There are these techniques called laser flash photolysis, stot flow, which are in my lab. to measure the oxygen binding, and we saw that this mutant is more or less similar, not much difference from wild type protein, and may be useful as a transport protein, oxygen carrier. So we published this paper also, and this was done in myoglobin, but you cannot use myoglobin for as an oxygen carrier. You need human hemoglobin, which we are trying. I am not showing that story today, maybe another day. But at that point, when we published this, people did not understand, okay, and they started taking out a lot of news in the media. New researchers developed blood substitute, and very Ubiquity, a lot of these very interesting ones. Subke kaam aayega ye khoon. But we did not make any khoon. But as you know, sometimes in media, the understanding is not there. We are making just a protein. Anyway, so to end here, I hope I have impressed upon you that it's very important to study hemoglobins. They are significant molecules. If you still don't believe me, hemoglobin is the only protein on which 2 movies have been made. Okay, we believe and trust in movies. In the Hollywood, 2 movies have been made with hemoglobin as the title, so they must be important. And credit goes to all my students who does this wonderful work while I do most of administration, nothing in the lab, a lot of funding agencies. And I end by saying, that the secret of life is to have a task, something you devote your entire life to, something you bring everything to every minute of the day for the rest of your life. And the most important thing is it must be something you cannot possibly do, challenging task. For me, it's all hemoglobins. Thank you very much. Thank you for taking a little extra time. Thank you.